Staphylococci are gram-positive cocci and they are observed as grape-like clusters. They are non-motile and non-sporing, approximately 1 micrometer in diameter. And this group of bacteria can be grown in ordinary culture media with a temperature range of 10 to 42 degrees Celsius, and the optimum is 37 degrees Celsius at a pH range of 7.4 to 7.6. And this group of bacteria are aerobes and facultative anaerobes. And in nutrient agar, they produce circular, smooth, convex, opaque colonies, and they are easily emulsifiable. And in blood agar, the colonies are very much similar to those present in the nutrient agar. In addition, they also shows beta hemolysis also. Then the McConkie agar, they produce very small and pink color colonies due to the lactose fermentation. And selective media also used to isolate the Staphylococcus from the samples like feces that contains other bacteria. And the Staphylococcus can tolerate 8 to 10% NaCl, then lithium bromide, then tellurate, then polymyxin. Addition of these compounds make them selective. Then the mannitol salt agar. It is both selective and indicator media that contain nutrient broth, 1% mannitol, then 7.5% NaCl, then phenol red as indicator. Then it produces yellow colored colonies in mannitol salt agar. Then liquid medium, we can use peptone water or nutrient broth. It forms the uniform turbidity in the tube. And come to the antigenic structures. Some group of Staphylococcus have the capsule that inhibit phagocytosis, and they are said to be the more virulent group. And they have peptidoglycan. It's a polysaccharide polymer that provides rigidity. And the teichoic acid, they are the major antigenic determinant in all strains. And the protein A, it's a cell wall component of most of the strains. And toxins and enzymes. Staphylococcus produce a number of toxins and enzymes. They have an important role in their virulency. And the first one is hemolysin. Then 4 antigenically distinct type of hemolysins are present. The first one, alpha-lysin, it is lytic to rabbit erythrocytes and less effective against human and sheep RBC. And the beta-lysin, which is lytic to sheep RBC and not in human and rabbit RBC. Then gamma-lysin, it is lytic to human, sheep and rabbit RBC. Then the delta-lysin, it is lytic to human, sheep and rabbit RBC. Then next toxin is leukocidin, also called as the Pantone Valentine toxin after the discoverers. Then they have 2 components, fast and slow, and they make damage to the polymorphonuclear leukocytes and macrophages. Then enterotoxin is very much associated with the staphylococcal food infections and they are resistant to the gut enzymes. Then among 8 types, that is A, B, C, C1, C2, C3, D, E and H, in most cases the A group is present. Then toxic shock syndrome toxin, that is TSST. Most TSST producing strains belongs to the bacteriophage group 1 and TSST type 1 producing strains frequently colonize the vagina. Then exfoliative or otherwise epidermolytic toxins. They have 2 types, that is A and B. Then enzymes, Staphylococcus produce number of enzymes out of which coagulase, phosphatase and deoxyribonucleases are very much related to their virulence. First one, coagulase. Then there are 8 types starting from A to H. Then most of the human strains produce type A. Then they have a property to clot human or rabbit plasma and they are heat liable. Also they require CRF, that is coagulase releasing factor for clotting. Then two types of coagulases are there, that is bound coagulase and free coagulase. The bound coagulase or clumping factor can be detected by the slide test. They form the clot after the addition of rabbit plasma along with sample. Then the tube coagulase tube test is used to determine the free coagulase, which is responsible for soluble fibrinogen in the rabbit plasma to form the insoluble clots.
Download Blulins now, redefining quality education.